Okay, in the previous lecture, we have discussed about the digestive system. That means we have discussed about the complete structure of alimentary canal and all the structural features of different organs of the digestive system. Fine. Now, today we will going to discuss about the physiology of digestion. That means how and where, in which particular part of digestive system digestion of different nutrients takes place. Now, since we have discussed in the previous class that our human alimentary canal consists of mouth, esophagus, stomach, small intestine and large intestine. So, from the very beginning, that is from the very proximal part, that is from the initial part mouth, the digestion starts. So, you can say digestion starts in mouth and digestion completes in small intestine. Okay. Now, that means the starting of this digestion process has been taken place in mouth itself and it completes at the duodenum part of small intestine. Now, how this digestion takes place in mouth? So, first of all, we will going to discuss about the digestion process in mouth. Now, we have already discussed that mouth includes our lips, that is surrounding our oral cavity or a buccal cavity. So, we can say that oral or buccal cavity includes saliva. That means it is including all teeth, tongue, palate, but in the cavity what exactly is secreting is saliva. So, the saliva which secrete is by salivary glands. And in the previous lecture only we have discussed about the three types of salivary gland, that is submaxillary gland, sublingual gland, parotid gland. These three type of salivary gland which is located in the mouth region, or you can say just below the lower jaw and below the ear also, these glands collectively secreting saliva fluid. And this saliva fluid actually contains various components which also we have discussed in the previous class, that is enzymes, salts, water. Sorry, not water. Now, enzymes are of two types, that is lysozyme and salivary amylase. This salivary amylase, also known as ptyalin, the another name of this enzyme is known as ptyalin. Now, the main enzyme for our concern is salivary amylase, that is ptyalin, which play a very important role in digestion process. Now, as we all know, we human being, generally our diet depends on cooked starch. Our diet is cooked starch. We do not eat raw starch, or we do not eat cellulose diet, cellulose food. So, we eat cooked starch, and this digestion of starch actually starts from mouth itself. Now, this starch, which is, we can say, a polymer of glucose. That means starch is a long chain of glucose molecule, its monomer unit is glucose. And what is glucose? We all know C6H12O6. This is glucose. When more than one glucose molecule combine in a chain, either in the branched form or the normal unbranched form, it will form a starch. Now, whenever the starch breaks or completely break down, the simplest unit of starch will be glucose molecule. So, whenever the starch digests completely, we get glucose at its end. Now, what happened? How the starch digestion takes place? Like the starch digestion starts in mouth, and this digestion of starch which starts in the mouth is helped or acted upon by the enzyme known as salivary amylase. Now, this starch, when salivary amylase acts on starch, So it do not break it completely. Why? Because starch is having two main component, amylose and amylopectin. That means it present in their branched form as well as unbranched form. So whenever the salivary amylase act on starch, it do not degrade completely, but it breaks up into its simpler units like maltose plus dextrins. Now what is maltose? Maltose consists of 2 glucose molecule. Maltose consists of 2 glucose molecule, and the another whole chain of glucose we can consider under the dextrins. So that means in mouth the digestion has been started, but it's not been completed here because starch is a complex structure. So it needs more and more action or more and more activity of enzyme in different organs or different stages to get degrade completely into the glucose molecules. So in mouth only and only the digestion of starch started, but the digestion of fats and protein has not been started yet. Now the second stage of digestive system comes is stomach, because in esophagus there occur no digestion, only and only the peristaltic movement is taking place in the esophagus, through which the food bolus is entering into the stomach from mouth. So esophagus is just helping in pushing down the food bolus in the stomach. The second stage of digestion is stomach. Now what happens in stomach? Mainly the stomach is having certain types of gastric glands which secrete gastric juices. These gastric juices includes various components, that is enzymes, HCl, mucus. Now enzymes are very important. We have mainly two important enzymes present in the gastric juices secreted in the stomach, that is protein digesting enzyme, pepsinogen and prorenin. Now this pepsinogen and prorenin, here double N, pepsinogen and prorenin. Now these two are the protein digesting enzyme which is present in their inactive form. This is inactive form of enzyme which becomes active whenever the digestion of protein required. Now the activation of these two enzymes requires acidic medium, that means the pH should be low of about 5 or 6, or you can say basically this enzymes activates at a low pH and in high acidic condition. So that is why stomach is releasing the HCl, that is hydrochloric acid, in the dilute form, and due to this HCl only the enzymes gets active, and then only the digestion of protein will going to start. So what happened in stomach part? This pepsinogen first will going to convert into pepsin by the action of HCl, that is hydrochloric acid. So pepsinogen is inactive form and pepsin is the active form of enzyme. In the same way, now this pepsin will going to act on protein molecule. Now protein is the polymer of amino acids. You can say protein's monomer unit is amino acids. So amino acids are the building block of protein. Whenever the protein digestion complete, at the end we should get amino acids. But again I am telling, like here also the digestion of protein started but not yet been completing here. So protein digest or simplifies, or you can say break up into its smaller units by the action of pepsin, that is proteose, and you can say peptones, small polypeptide chains. Now in the same way, another enzyme, that is prorenin, by the action of HCl it will convert into renin. What is this prorenin, renin? These are the milk protein digesting enzyme. This will milk protein digesting enzyme. That means it acts only on milk protein, not any other protein. So this renin will going to act on milk protein, that is casein. Casein is a milk protein. This casein will going to convert into para-casein by the action of this renin enzyme. Fine. So this para-casein is the smaller unit or the smallest polypeptide chain of casein by the action of renin enzyme. So these two type of protein digesting enzyme in the presence of HCl will going to act on different type of protein, and it will going to convert these protein into their simplest unit, but digestion is not yet completed here. Now in stomach it is not like that ki only the protein digestion takes place. In a very small amount the digestion of carbohydrate or lipids also takes place, but it's of negligible amount. Fine. So maximum digestion of which nutrient takes place in stomach, it is of protein. Now this HCl is also secreting in stomach only and only for the activation of the protein digesting enzyme, because the protein digesting enzyme requires the acidic medium. Although this acidic medium is harmful for human organs also, so the stomach is the only area where the pH found to be low due to secretion of HCl, or you can say the acidic condition found only and only in the stomach region. But again we have the defense mechanism also to protect the wall of stomach from action of HCl, that is mucus. This mucus secreted by another type of specialized cells present on the wall of stomach, that is mucus secreting cells. This mucus is a very it's of very lubricative nature, and it actually prevent the action of HCl. That means it prevents the harmful action of HCl and protect the wall of stomach from being degraded or from being destroyed by this HCl. So if HCl is secreting in the process of digestion of protein, another pathway, that is mucus, is also secreting to inhibit, or you can say to suppress the harmful action of HCl as well. Now the third stage, these protein digestion which starts in stomach but not completed. Now the digested food, that means in which the protein somewhat has been digested, and you can say the starch which has been somewhat digested. So all these digested nutrient or fluid which transfer from stomach to the small intestine is known as chyle. This is chyle. Chyle is the digested food in acidic medium from stomach. Now it enters to small intestine. So the third stage for the digestion is small intestine, and small intestine maybe the most important is the region of small intestine is duodenum, where the digestion is going to complete now. Now when this chyle having an acidic medium or having acidic condition, when it enters into the duodenum, it can harm the wall of small intestine or the wall of duodenum. Now what shall be done in order to protect the wall of duodenum from the acidic action of this chyle? So for this we have another secretion, another fluid, or you can say another juice that is secreted by the liver, which is known as bile juice. This bile juice actually contains, bile juice is actually producing in the liver part. This bile juice through hepatic duct passes to gallbladder and remain stored there. So bile juice forms in liver, but it transfer to the gallbladder for storage through a duct known as hepatic duct. These are the duct comes out from liver. That is why it is known as hepatic duct, because the term hepatic is used for liver. Now, for some time, the bile juice remains stored in gallbladder until and unless it is being used for the process of digestion. Although bile juice does not contain any enzyme, it does not have any digestive enzyme, but it contains two main components, bile salts and bile pigments. Bile pigments has no important role in the process of digestion, but it will going to provide color to the urine and to the fecal matter. So our urine color and the color of fecal matter is due to these bile pigments only. Bile salts are the important component. These salts are alkaline in nature and will going to neutralize the acidic condition of chyle. So this bile salt is going to neutralize the acidic condition of chyle, which is coming from stomach. So that is why bile salt is very, very helpful. As well as this bile salts is also going to help in the process of fat digestion. So later we will going to discuss it. Now this bile juice which remains stored in gallbladder, now it will going to secrete into the duodenum through the duct known as hepatopancreatic duct. Why? Because this is the common duct through which bile juice from gallbladder and the pancreatic juices from pancreas get mixed up and secreted into the duodenum. Now this duodenum, which is containing the digested food which has entered from stomach, it include all those digested nutrient which is incompletely digested, like for example by starch digestion we have got the maltose and dextrin, and by protein we have got protease and peptone. Now it further need to be digested, which is going to complete at this duodenum stage. Now what happens at duodenum? Both type of juices secreted, that is bile juice and pancreatic juice. Fine. Bile and pancreatic juices will going to secrete. Now this bile juice which is having the bile salts is going to help in the digestion, and pancreatic juices also contains various types of enzymes, that is trypsinogen, chymotrypsinogen, pancreatic lipase, pancreatic amylase, and nucleases also, that means which will going to digest the nucleic acids. Trypsinogen and chymotrypsinogens are the protein digesting enzymes. Pancreatic lipase is the lipid or fat digesting enzymes. Pancreatic amylase is the carbohydrate digesting enzyme, that means the starch digesting enzyme. Now how it going to act? Now before the digestion, like for example we have discussed the starch. After digestion of starch in mouth we got dextrose, dextrin and maltose. From starch digestion. Now this pancreatic amylase will going to act on it and will going to give different products like glucose plus another molecules of maltose plus limit dextrins, right? That means different or simpler molecule हमें मिलते हैं, like dextrin से हमें glucose, maltose से भी we have got glucose, but another dextrin, since it is a long chain of glucose, so we must have get the maltose from it and also the limit dextrin, that means the more smaller unit of smaller chain of glucose molecules. The another action is of, for example, trypsinogen and chymotrypsinogen. Now this is again the inactive form. Chymotrypsinogen first will going to convert into chymotrypsin. Fine. This chymotrypsin is an active form of protein digesting enzyme, and this chymotrypsin will activate by the another enzyme known as enterokinase. This chymotrypsin will then act on trypsinogen and convert into its active form known as trypsin. Now this trypsin is the protein digesting enzyme, will then going to act on those proteose plus peptones and will convert into smaller polypeptides, or you can say polypeptide means which include the dipeptide, tripeptides and polypeptides. So another unit or the more smaller chains of amino acid we have got after the digestion of proteoses and peptones by the action of the protein digesting enzyme trypsin. Now after that the digestion of fat is there. So we have pancreatic lipase plus bile salts. Now the digestion of fat is difficult because it is of oily nature, and the human body is actually having the fluid mainly of water. That means we have major amount of water. So our enzyme is the nature to act on the water soluble component. So it is very important to make the oily nature of fat to become water soluble, so that enzyme can easily act on it. So this fats need to be emulsify. So this emulsification process has to be done. What do you mean by emulsification is? Emulsification is the breakdown of large molecule of fat into small globules, or you can say small droplets of fats. Fine, that means the smaller molecules of fats is to be formed through the process of emulsification, and this emulsification is done by bile salts. This is done by bile salts. Now after that, this emulsified fat is then acted upon by the enzyme known as lipase, because now they become water soluble and it is more easier for the enzyme to act on the small droplets of fats rather than to act on the large complex molecule of fats. Now this lipase, when act on it, now it will going to degrade into the form known as triglycerides. Triglycerides is the component of 3 fatty acid plus 1 glycerol. So whenever the digestion of fat complete, we should get fatty acid and glycerol molecule. So here we have discussed about the digestion of protein, starch and the fats. Now these are the enzyme which we have discussed about is of the enzyme we have got from pancreatic juices. Now the intestine, that means the small intestine, also have their own juices, also have their own digestive enzymes, and the secretion of small intestine itself is collectively known as succus entericus. Collectively known as succus entericus. Now after secretion of the pancreatic juices and the bile juices in the duodenum, when the juices of pancreas and liver, when they are acting on the components of food, so later on whatever the food component remain left need to be digested is then again acted upon by the digestive juices secreted itself by small intestine, that is succus entericus. Now this juice also contains various enzymes, like we can say intestinal lipase, intestinal amylase, we can say proteases, protein digesting enzyme, and you can say nucleases, nucleic acid digesting enzymes. So these enzymes will then, now these enzymes will going to act on all those digested nutrient which has been digested upon by the pancreatic enzymes. Now for example, the intestinal amylase will going to act upon maltose plus dextrins and completely break them into glucose. So we have finally got the smallest unit of the star, that is glucose. Same the lipase. We already have got the triglyceride after digestion of fats by the action of pancreatic lipase. Now the triglyceride will going to digested by intestinal lipase, and we have got from here 3 fatty acid and 1 glycerol. So here also the complete digestion of fat has been taken place. Third, the protein. So we have already got the proteoses plus polypeptides. So after digestion of the enzyme proteases, now we have got the smallest unit, amino acids. Fine. So in this manner, the simplest substances we have got from the digestion of protein, carbohydrate and fat. That means that is where at the duodenum the digestion of food completes. Now the main important thing about fat is that fat digestion need was emulsification process, that is an extra effort has been required for the digestion of fat which has not been required for the digestion of carbohydrate or protein. So this is an extra effort for digestion of fat only, that is emulsification need to be done by the action of bile salts which is coming out from the bile juices, and the bile juice is actually prepared at liver. So at the duodenum, the digestion of food has been completed in the duodenum, that means that at the distal end of the duodenum part, the digestion of food is completed now. Now in the jejunum and ileum part, the absorption of food is going to start. Once the digestion completes, the next process, that is the digestion, after digestion the absorption is going to start. Now what do you mean by absorption? That means whenever the digested nutrients transfer from digestive system to blood, and this blood will going to take all these digested nutrients to different tissues or different cells. So absorption is going to start in small intestine again, and the small intestine, the main part is jejunum, and it will going to end again in the small intestine, that is in the ileum part, you can say, right. So absorption starts in the small intestine and it also ends up in the small intestine, but here I am talking about the absorption of only digested nutrient, not water. So the digested nutrient is going to be absorbed by jejunum and is going to be absorbed by the blood from jejunum and the ileum part. Now absorption process also need various mechanism, like absorption can be done either by passive transport or by active transport. Fine. Now what do you mean by passive and active? Passive means which can include simple diffusion, or facilitated diffusion. That means in passive transport there is no energy expenditure occurs. Here osmosis also there for water. For water absorption we need osmosis. Fine. Now active transport, here the energy expenditure takes place. In active transport energy expenditure is required, and as we all know, the passive and active transport means passive where the movement of substances takes place along the concentration gradient, while in active transport the movement of substances takes place against the concentration gradient. So that is why passive transport do not require the expenditure of energy because it is occurring along the gradient, while active transport do require expenditure of energy in the form of ATP because it has to be transport against the concentration gradient. So as we all know that passive transport, obviously it must include different type of nutrient as well as water itself, for through osmosis the transportation or the absorption of substances takes place through passively. But most important nutrient, for example glucose or the other important digested nutrient, it also may transport through actively because it is a very important nutrient which need to be transported actively also, even if the concentration is lower in the digestive system and more in the blood, then also it keeps on absorbing. That means against the concentration gradient also it can absorb by the expenditure of energy because it is important digestive nutrient and our body need glucose at a very high level also. So that is why it needs active transports. So this is all about the absorption process which is started in the small intestine and also get completed in the small intestine, but here we have discussed about the absorption of digested nutrient. So this is up to this lecture we will going to discuss the absorption of digested nutrient. On the next lecture we will going to discuss about the absorption of water. Thank you.